Okay, I think I'm going to kick it off. It looks like our numbers aren't coming in fast and furious anymore, but again, welcome to our very first Darwin's Cat Science Talk. I'm Jill Simmons, Darwin's ARC CEO, and I'm so pleased for you to join us today for a conversation with our scientists. As you likely know, Darwin's ARC is a nonprofit, and we were founded by pet-loving researchers who believe that our love for our pets is an untapped scientific resource, and that all of us who share our homes and our lives with pets and cats in particular for today know them better than anyone else and care probably the most about their health and well-being. And what that means is that Darwin's cats and Darwin's ARC, really pets and their people are the beating heart of who we are. And we're partnering with all of you to gather the data that's needed to advance groundbreaking research, which you'll hear a lot more about in a minute. I know everyone joining us here today shares this belief because you all are our most committed community scientists. Folks who've taken all 19 Darwin's Cat surveys, folks who've enrolled your cats in our DNA sequencing research, folks who've done both. Thank you so much. I've been incredibly inspired by the commitment and the passion that everyone has brought to our research and to your engagement with our work. I'm impressed, but I also understand it. I am a lifelong cat lover and an enthusiastic Darwin's Cats community scientist myself. I've had a cat in my life since the age of 5, constantly, and I know that I'll have a cat in my life for the rest of my days. My cat, Gus, now is a proud member of the DNA sequencing research, and I'll confess to sneaking him into today's presentation. So look for him on the slide at the start of Chad's presentation. He's there getting brushed with our comb. But before I hand it off to the folks that you came here to hear, I'd like to go over a few logistics about how to ask questions for our Q&A. Most of you are probably familiar with Zoom webinars, but there is a control bar at the bottom. If it disappears, just bring your mouse down there and it should reappear. There's a box or an icon with the Q&A button. When you click on that, there should be a space to ask, to type in your question, and that's how we'll be moderating our questions. As you can see, we have 113 folks, 14 now, on today's call. So we aren't able to answer questions live, but we are collecting them all. We received actually nearly 200 questions from all of you just during the registration process, and I'm certain we're going to get many, many more today. So I need to apologize in advance. We're not going to answer every question, but that doesn't mean they're going into a black hole. We will be keeping them all and are really looking for other ways and thinking about how we can answer questions in our newsletter, our blog, in future science talks. So please ask your questions and know that even if we don't get to it today, we're keeping track and we're thinking about the themes that would help that you're interested in and that you would want to hear more about. So with that, I'm gonna, I said I'm a cat lover to my core, but one thing that I am not is a geneticist. So it's my great pleasure to introduce Elinor Karlsson, Darwin's ARC co-founder and chief scientist, who is also a geneticist and computational biologist. So Elinor, take it away. And I will hopefully advance the screen. Well, okay, I got to start right now because I'm driving the slides and I want to apologize in advance. This is not one of my core competencies, so please bear with us as we try and navigate all of the technology. All right. And Elinor.
Thank you very much, Jill. And thank you to everybody for being here. We're really excited to actually have a chance to talk with you guys. You can't imagine how much excitement there is every time, Kasia comes in the morning and reports how many people have signed up and whether we've got new DNA kits ordered. So it's great to actually be able to tell you more about what we're doing. So let's see. We've just figured out changing thing and it's not quite working yet, so we're going to give it just a moment. There we go. Okay, it turns out that I can actually click and advance the slides, which is great. So Darwin's Ark actually started as a dog research project when I started my lab at the UMass Chan Medical School in 2014. So I'm, as Jill mentioned, a geneticist, and I'd been studying dogs as part of my PhD for many years. And one of the things I've learned was, one, that all of our studies were too small, and two, every single time I talked to anybody about what I did, if they owned a dog, the first thing they would do would be to pull out their phone, show me a picture of their dog, and start telling me all about them. And so I was sort of thinking, what should I do with my new research lab that I'm starting up? And I was like, maybe there's a way to actually engage with people to get all of that data that they keep on giving me when they talk to me and put it into a form that would be useful for our science. And so that was sort of the vision. We wanted to build a big resource where we could actually capture all this information from people that had dogs. And we also wanted it to be an open data project. I figured that people weren't going to want to participate just to support our own research. They wanted to participate to help all research. And at the time, part of the reason why our studies were all too small is that every scientist was sort of doing their own thing. Then reality hit. It turned out that lots of people did want to participate, more people than we could actually handle as a small research lab. And so together with Charlie Liu, we actually started the nonprofit Darwin's Ark. Down in the corner there is Brittany handling all of our dog saliva swabs, which is how we collect DNA from dogs. There were an awful lot of them. It was a big challenge for us. Okay, so Jill, I'm still having trouble advancing this. There we go. Okay. So science, what is DNA? So DNA is basically the instructions for how to build an organism, a human, a dog, a cat, and tell it how to keep running. So it's got all sorts of instructions, and they're not just about how to, develop a human or a dog or a cat, but also what to do when things happen, like you get exposed to an infectious disease. Your DNA sort of in each of your cells has sort of the building blocks for what you do in that situation. You inherit your genome or your DNA from your parents. You get one copy from your mother, one copy from your father, and those two copies together make up your genome. And the genome is basically just all your DNA. So if you think about whenever you see DNA, it's shown as this like helical thing, like this double helix thing. So each one of those helixes is basically a whole series of 4 different chemicals stacked on top of one another to build up the sides of that ladder. Each of those has a very complicated name. We tend to abbreviate them as A, C, G, and T. And so when we write out DNA, we just write it out as a long string of A, C's, G's, and T's, which is basically telling you the way those instructions are written into the genome. We've gotten really good at sequencing DNA, figuring out the exact order of the letters as we go along a strand. At the Broad Institute Genomics platform, for example, they have lots of machines, and they can read billions and billions of letters or bases every single minute. The problem that we have is we don't actually know how to read the DNA. We don't know what it's telling us about how cells work and where diseases come from and things like that. And so that's basically what our research is about. And the other problem, of course, is there's a lot of it. The human genome, that long string of A, C's, G's, and T's, is about 3.1 billion bases or letters long. In dogs, it's about 2.4 billion bases. Cats are a little bit bigger at 2.7 billion, but we basically say all mammals are sort of about the same size. Birds are nice, tidy 1 billion bases. And I like to tell when I talk to students is that if anybody ever asks you to work on axolotls, you should probably run in the other direction because their genomes are 30 billion bases long, and we're not totally sure why. Okay, so what Darwin's Ark is about is trying to figure out what that DNA is doing. It's one of the biggest problems in medical research today is just knowing when a change in DNA actually leads to a change in either a trait or a health status. And I find it to be a really fascinating question. I remember thinking years ago, that you have these dogs that are completely obsessed with retrieving balls, and then you have other dogs that could care less about retrieving a ball. And somehow, somewhere in their DNA, there's a change in that sequence of A, C's, G's, and T's that creates a difference in how interested an animal is in retrieving a ball. And that's just absolutely on a scientific level, really fascinating to me. Like, how does that even work? And then also from a sort of like understanding disease perspective, if you think about it, you've got things like retrieving, but you also have things like compulsive disorders, where somewhere in their DNA is a change. And if we could figure out what it is, we might be able to understand where diseases are coming from and find new and more effective ways of actually treating the causes rather than the symptoms. So this is what Darwin's Ark is designed for. We collect, so if you think about us, and so I often get asked, how is it different to participate with us versus participating with, say, sending your DNA off to a company to look at your dog's DNA and tell you something about it? So Darwin's Ark is actually a very different kind of thing. When you send off your sample to a company to get looked at, they basically take everything that is known about dog DNA and report it back to you. What we're doing is the opposite. We're saying we want to find the things that nobody knows yet. We want to discover what causes retrieving or what causes cancer. These are really big problems, and so we need to collect a lot of data to do it. So for every single animal, we collect a huge amount of genetic information, 100 to 200 times more than what you would do if you were actually sending it off to a company just to find out the breed and whatever. We use a different approach. They do something called genotyping, which looks at a small number of positions in the genome. Small in genomics land is like 100,000, versus we actually go across the entire genome and look at all of the bases that an animal has, because we don't know which ones are important yet. We also want to know a lot about each animal. We want to know whether they're interested in retrieving a ball, or what diseases they get, or even what they look like. And we want to actually find out what changes as animals get older. Why do some animals live longer than others? What diseases do they develop? And we don't own the data. It's an open data project. We share it openly with other scientists when they come and talk to us and ask for it. We want to make sure that as many people are using it as possible, because that is the best way to make science happen quickly. And the final piece, and this is something that why we're here today, and something that we're trying to get better and better at over time, is that rather than just getting you to give us information, we want it to be a conversation. As we discover things, we want to be able to tell you what it is that we've discovered and what it means for your pet. So how do we do this? So I'm talking about dogs now because that's where most of our research to date has been done. The cat project, as everybody on this call knows, is very new, so we haven't got a lot of data yet. But we're basically, all this stuff applies to cats as well, which is why I'm talking about it. So how do we connect a change in DNA, which we call a genotype, to a change in a trait in an animal, which is what we call the phenotype, what the animal looks like, how they behave, what their diseases they're getting. So the first step is we find some individuals that differ in some way. So I've got on the right here a beautiful picture of Nellie the Beagle, who could care less about retrieving a ball, and Besco, who was actually pretty good at retrieving balls. She wasn't as excited as some dogs, but she was pretty into it. We get dogs that have different traits and that are different scores on a particular trait. We sequence their DNA, and then we compare the DNA of dogs that really love retrieving to the DNA of dogs who don't like retrieving, and see where their DNA is different. And that tells us something about where it might come from. The thing that really complicates this is that it's not that easy. It's never that easy, of course. So if you're talking about something like a behavior, there could be dozens or hundreds of different genetic changes involved in influencing that particular trait in dogs. And so we need to be able to find many different changes that each subtly influence the trait. And then we also know that things change based on a dog's life experience that might have nothing to do with the genetics. And so we have to take that into account too. So we found out from dogs it works well. This is one of our simple traits. So a simple trait is a trait that's controlled mostly by changes in a single gene. And so this is our trait about, is your dog brindle? We asked a whole lot of people what color their dog was. Some of them reported that their dog had this brindle stripy coat pattern. We fed all of the DNA from dogs that were brindle and dogs that weren't brindle into the software and said, look across the entire genome, all of their DNA. So that's going across the bottom of this plot, going from the first chromosome to the 38th chromosome in dogs. And so look at how different those two groups of dogs are. And I think everybody that is looking at the screen can probably tell that we were really good at finding the position that actually influences brindle. We definitely found that gene. That is an incredibly big peak. It gets much harder with complex traits like retrieving a ball. And once again, I want to, people sometimes ask, how do the surveys feed into this? The only reason we can do this work is because of the survey data. Getting the genetics is just one piece of it. We need to know about the animals. And so in this case, we asked a whole lot of people how interested their dog was in retrieving a ball. And we got data here from about 3,000 dogs. And we did the same thing. We said, let's compare the dogs that are less interested to the dogs that are more interested, go across their whole genome from chromosome 1 to chromosome 38 and see how different they are. And there's two things you can see. One, this is a complex trait. The peaks are not as high, and we have many of them. But the other thing that we can actually see when we look a little bit closer is that underneath our top peaks in this particular, what we call a GWAS study, we looked to see which genes were there. Each of the top peaks actually had a gene in it, which was pretty cool. And when we actually looked at what those genes did, every single one of them was actually connected to something to do with brain function and specifically behavior and learning. And so it really suggests we're on the right track to finding the changes that actually influence this trait. We can also detect when it's not genetics and it's something else entirely. So we have this in the dog project, we had a trait we measured that we called toy-directed motor patterns, which was basically a bunch of questions about how much your dog liked to play with toys. And when we looked at our data, what we discovered, possibly unsurprisingly, is that this is not just a genetic trait. This is also a trait that's influenced by age. And I like to tell people that, yes, what we figured out was that puppies really like to play with toys. And also that as you get older, you tend to be less interested in playing with toys. Okay, so we did all of this work. It was a lot of fun. We actually were able to ask, basically question a lot of things that everybody thought they knew. So we did we welcomed all the dogs, and this is not just single breed dogs. And we looked at all the pet dogs. And one of the surprising things that came out of it was that it turns out that breed of dogs actually predicts very little of their behavior or their personality. There's a little bit of an influence, but it is really difficult to pick out. So there's a lot of what we call sort of confirmation bias when people think that they see these really dramatic differences. The other thing that came out in all the press coverage we got of this was actually the fact that I have never owned a dog. I've done dog genetics work my entire career, but I've never owned a dog, and there's a reason we called it Darwin's Ark and not Darwin's dogs. So I am a total cat person. This is my cats Lacey and Beagle on the left. Not only am I a total cat person, I am the kind of cat person that owns a giant cat wheel for my cats to run on. And at this point in time, the closest I've actually gotten to owning a dog is probably that straw dog that my cat Hopper is hanging out with there. Not only that, I come from a family of people that are completely into cats. I texted my siblings earlier this week for pictures of their cats, and this is what I got. And there's only one dog that snuck in there in the bottom left-hand corner there. So one of the things that's been really interesting for me as a person that likes cats is to understand how cat genetics introduces new challenges to all of this work. So in dogs, we actually had a bit of a head start. There was already decades of genetic studies that have been done in dogs. Dog genetics is a bit simpler because of this weird thing where most of the dogs are either a breed or a mix of breeds. We had something that people wanted to know about their dogs that we could actually already do from genetics, which is return their dog's breed mix. And we knew more about which traits were actually influenced by genetics, which ones were heritable and which ones tended to be not heritable. So we had an idea of where to start looking. In cats, there's been very few genetic studies. They tend to be very small. Cats are, for the most part, don't have breed ancestry. There's a few cat breeds out there, but mostly cats don't have breed ancestry, and their ancestry is probably a lot more like humans than it is like dogs. It has more to do with where they came from originally than any kind of breed. We can't tell people very much initially because we don't understand a lot about cat genetics yet, and we don't actually really even know which of the traits and diseases are actually influenced by genetics and which ones are just environmental. And actually, for those of you that have asked, is the survey data alone important? This is where it really comes into it, because it's a way for us to start figuring out what questions we should be asking and how cats actually differ from one another in terms of the survey data is going to be really interesting to see. Okay, so what are we trying to build with Darwin's cats? I'm just going to end here by saying our goal is to take these animals that I absolutely love and you guys do as well, but that actually we don't know very much about right now. They're sort of a black box when it comes to a lot of things, both in veterinary medicine and in science, and understand them much better in a way that we can actually help make their lives better. On the right there, I have my two first cats, Leif and Darwin, who are both ginger cats, which is clearly proof that ginger cats are absolutely the best cats and obviously amazing. But yeah, so what we're trying to do is build the largest cat science community. Big data sets are essential for understanding complex traits like diseases. We want to build open data resources for cat research. We want to engage with all the other people out there working in cats. And finally, what we've really been focused on recently that Chad's going to talk about next is what we've been doing to develop new methods to actually do genetics in cats that actually addresses the fact that they're not dogs. And in order to engage them in research, we need to do things in a way that's more cat appropriate. So thank you very much. And I'm now going to hand it off to you, Chad.
Okay. Thank you, Elinor. I'm Chad Nusbaum. I've been in the DNA sequencing technology business since the early days of the Human Genome Project back in the 90s. Elinor told you about the initial project that was done with dogs, and the DNA for that project largely came from cheek swabs. Some of it, I think, came from blood. We were concerned that cats might not be as cooperative with that kind of collection, and we wanted to make sure we were using a method. Since everything's based on what we can learn from looking at DNA, we wanted to make sure we had a method of getting DNA from cats that was as easy as possible. Getting blood requires a veterinarian. Getting saliva DNA requires sticking a Q-tip in an animal's mouth and rubbing against its cheek for 30 seconds. I wasn't sure, we weren't sure that cats would be as cooperative for that. And we reasoned that we might be able to do this from fur. And I sort of got involved with the project over a conversation about whether we can do this from fur. And I said, well, nobody does it, but it should be pretty doable. There we go. So if we're going to do a DNA sequencing method or any method for anything, we first want to ask ourselves, what does it have to do? We call this requirements definition. So the requirements of a DNA sequencing method for the cat project would be, first, that it's not a problem for cats, so we don't want to stick needles into cats or stick things in their mouths. It has to be easy for the cat parent to do, since we're asking all of you to do the collection from your animals, and some of you have done this already. We want to be able to get DNA of a sufficient quality to answer the questions that we want to ask. It can't be too expensive, since we're a nonprofit, a community organization. And it's scalable, and what scalable means is we have to do thousands of them. We're planning on doing at least 5,000 of these within the next year and a half. So I sat down with a really talented molecular biologist I used to work with at Broad back in the teens. Oh, nope. That's the slide after this one. So let's go through the logic of this. Pluses and minuses of working with fur. It's a plus that fur is easy to get. Animals are loaded with DNA, and it seemed likely that some of that DNA was going to show up in the fur. We were concerned, and there was a lot of concern in the field, that the amount of DNA in hair would be small and that the DNA would be badly damaged. There were existing methods for extracting DNA from hair. These were largely developed for either forensic samples like you see on NCIS, where they have one hair and they catch the mass murderer, or for museum samples like you have a little hunk of hair from a quagga that's been sitting in the museum for 300 years and you want to be able to extract DNA in that from sequencing. And usually the amount of DNA in those samples is very low and the quality is poor. And the question is, can we develop an easy method that's going to give us the amount and quality of DNA that we need? And so now I'm sitting down. Oh, yes. Yes is the cut to the chase here. The short answer is yes, and I'll tell you how we got there. This fellow in the second photo is Benoit Molony, the talented molecular biologist. And he and I sat down and spent an afternoon talking about how would we do this? What are the steps we have to take? And how can we make this work in a way that's affordable and gives us what we need? So those of you who have done this already will recognize what's in the left-hand picture, that's a standard 15 mil laboratory tube. And the pet owner has inserted into that a comb that's combed their cat. and caught a little wad of hair. This thing goes into the laboratory process. And so when we do extraction, that is getting DNA out of the fur, we add a little enzyme solution that partially dissolves the hair, the fur, and so it leaches into the liquid. And then we want to prepare that DNA, which means cleaning it up and modifying it so that it can be read by a DNA sequencing machine. That's what's happening in this middle picture. And this is your standard laboratory process of handling 96 samples at a time in a plate. So we're handling large numbers at a time. That's me using an 8-channel pipette, so it goes very quickly. And what's happening here is a number of washes where we remove the protein from the hair, any other junk that's floating around that's not DNA, and then we modify it so that when we squirt it into the sequencer machines that you see in the last picture, the machines know what to do with it and can read it out very cleanly. And we also mark them so we know which DNA came from which sample. And then once those DNA sequences are delivered to the computer systems, those will be picked up by Kasia, who will tell you in the next section how analysis of them is done. So we tried this on 100 samples. And the question we asked ourselves, so we always ask ourselves after a pilot like that, is how did we do? This is a new method. And how did it perform relative to the requirements that we set in the earlier slide? So first of all, it worked very well for cats. Nobody complained about doing this. It was very easy. Some of the cats even seemed to like it. We got DNA at a quality that we were surprised at. It was much better yield of DNA. In fact, at first we were getting too much DNA and we had to dial things down. And virtually every sample succeeded through the analysis phase. The other sort of bonus here was that since we were starting with fur rather than saliva, and saliva, you know, the mouths of mammals and animals in general are full of bacteria. When we sequence the DNA from the saliva, you get the animal, but you also get all the mouth bacteria. And so about 20% of the sequences that came from cheek swabs were bacteria. We were getting less than 1% bacteria, so it's a little more bang for your buck since DNA sequencing is one of the expensive steps here. We were able to make it affordable. We simplified a lot of the steps in the process. I think we may be able to make it even more affordable, but it's not more expensive than saliva, so that would have been an awkward point if it was. And since we were able to do 100 in a batch, we can easily do batches of 100, so it scales very nicely. We've got 500 cats underway right now, aiming to get 5,000 by the end of next year. So where do we go from here? One of the questions we've been asked a lot is, can we sequence DNA from deceased cats? Because there are a lot of deceased samples that people have kept, and it would be valuable because these are the animals come with the same kind of health history and behavioral history that current animals have. And so in the most recent batch that we did, we included 3 deceased cats, including one that was 5 years old. In each case, we got good enough DNA to proceed. So we'll have to do a little more optimization on this because it looks like the amount of DNA you get is a little bit less. But I think we're very optimistic that we're going to be able to do this easily. The other thing that I think is worth noting here is that fur or hair is a general attribute of mammals. So this could present a general method for doing genetic analysis of any mammal, including animals where sample collection is extremely difficult, like protected or endangered animals. In fact, I recently had a great conversation about this exact problem with someone from the zoo here in Boston who I met at a meeting, and she was talking about how difficult it was to get samples from apes because you know, they only get a health exam every other year, and only if they're really healthy will they even let you collect some blood. And so, you know, apes and endangered animals in general are highly regulated. And so I asked, well, can you get hair from these animals? And she said, oh, yeah, they leave it all over the place. I said, why don't you start saving those hair samples? Because this is going to be an opportunity, and it'll really open up that field as well. So we see a lot of opportunity going forward once we've gotten it going in cats. Now I'm going to hand it off to Kasia, who is going to tell you about how we actually start to learn from these data.
Great. Thank you, Chad. So, now that we have technology we are excited to use to collect genetic information, let's take a step back. What about the flip side? Because genetic data in isolation can only give you a small part of the picture. To really start to dig into understanding cats, we need to collect phenotypes. So basically observable information about the cats. So I'm Kasia Bryc, Director of Research and Scientific Engagement at Darwin's Ark. And, what I've been really focused on is maximizing the potential discoveries that we can make out of everyone's contributions here. So if your cats are part of these almost 14,000 cats that have joined Darwin's Ark, then you know that we're asking you a whole lot more about your cats than just their genetic information. This is an amazing number of cats. It's just been so exciting to be part of this, so much fun. We're excited to, of course, double or triple this number. But we already have an amazing, amazing group of cats. So let me take a moment to introduce you to Darwin's cats. We have cats that are born all over the world. That's what this map is showing, and all over the US. So as a scientist, this is really exciting to me because we want to paint a picture of domestic cats from all around the world. And so unlike a lot of clinic or university-based studies that sort of collect cats in one location, the Darwin's Ark model allows us to capture a diverse, if slightly US biased, set of cats to study. We have, let me see if I can advance my slide. We have young cats. We have old cats. We have cats from cities and from the countryside. We've got indoor cats, we've got outdoor cats. You know that we collect information about what your cat looks like. So we have short hair cats, long hair cats, tabby cats, non-tabby cats. We have almost 1 million survey responses. That is an amazing amount of data. And as a scientist, I am so thrilled because this is the scale of data that really starts to allow us to estimate rates of these traits at a resolution never before seen. So we can, for example, have enough data to start to say, how common are orange cats? You've told us about the health of your cats. So you've told us about their body condition, diseases that affect them. This information is going to be crucial for us to start understanding not just the genetics of these conditions, but hopefully also learning about how their environment, their exposures might affect the likelihood of cats developing these conditions. So what are we doing with all this information right now? Well, for one thing, we can combine all these survey answers, especially for cats that we have complete survey information for, to start to learn about patterns of shared, what we refer to as behavior factors, meaning personality differences that are variable among cats. So for example, similar to what Elinor talked about in dogs, there is a factor that captures your cat's interest in play, especially with toys. Another factor we're picking up on in cats is how much a cat enjoys people's company and how sociable they are. These behavior factors combine information across sort of a large number of survey questions and derive a sort of underlying estimate of where a cat falls on this spectrum. This is great because with data this large, we can start to understand how cats do and don't vary, and for example, how they change over time and how these personalities might be shaped by cats' unique home exposures or environment, and eventually genetics. But for now, we can actually get insights from just the survey data alone. So if we look at that first play behavior factor, we can see that as with dogs, now we know cats likewise become a bit less enthusiastic about toys and playing chase as they get older. This might not be a surprise to anyone on this webinar, but it is wonderful validation that our surveys are doing a great job capturing these components just from your survey answers. And this is the kind of insight that we can only get when we have both, first of all, large numbers of cats, but also with large numbers of survey answers on each cat. So we can start to explore the interactions between these behaviors and other covariates. And eventually we hope to lead into genetics, right? And so as Elinor discussed, we're trying to grow a body of information about the genetics of cats. And we can start to build this out first by understanding cat genetic diversity. So how are cats from different places and breeds related to each other? And once we understand this population structure of cats, we can start to investigate the genetics that underlies simple traits, such as physical traits that are strongly driven by just one or a few genetic variants. And then as we get larger and larger sample sizes, we hope to then be able to explore more complex traits or diseases that might be shaped by hundreds or thousands or even tens of thousands of genetic variants. Many of you have supported our genetic research. Thank you. And you sent in perhaps a little bag or a tube with fur in it. We have started sequencing some of those early participants. In the first quarter of 2025, we were really focused on getting the fur sequencing working very well that Chad just presented on. And you heard about those successes, so we're really excited about that. And we were gearing up to start sequencing the rest, but hit some bad luck due to turmoil in the scientific community. We lost our sequencing lab partner. The good news is that we are starting to work with a new partner and expect to restart sequencing by July, which hopefully will keep us on track with our goal of 1,000 sequences by September. We are starting to get some very early results. This plot shows our sequenced cats. So each cat is a dot on this plot. And it's shown by their overall genetics. So cats that are close together on this plot are genetically more similar, and cats that are farther apart are most different. And we're looking at these largely US-born cats, and the largest driver of differences among these cats seems to correspond to geography. So cats from the West Coast versus cats from the East Coast seem to be separated from each other, as well as the South and the Midwest. This is quite different from what we see in dogs, because in dogs, they tend to be most similar to other dogs from the same breed, regardless of their geographic location. So that was a really complicated way to show that cats are not dogs and their genetics are not the same, but that's what's really exciting. And I think this has never been published before. So we're already making exciting discoveries. And what's next? Well, we're still on track to hopefully sequence those 1,000 cats this year. And we need those large numbers of cats from different regions, from different breeds, to have power to detect these ancestry differences so that we can start to build a more detailed picture of cat history and ancestry. Once we have a good scope of that, we're excited to look for these genetic associations. So what regions of the genome of cats are connected to traits or behavior or health? I will make a plug that all the data that you have answered so far is incredibly valuable to power this research. And answering all the surveys is really helpful, especially please, 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 if you are participating in genetic research. And of course, the more cats that we have genetic information for, then the better our power to find these associations. And hopefully the genetic variants that impact our cats and make them who they are. So with that, I just want to say, as a research scientist, I am just profoundly appreciative of everyone here for their support of this effort. It really, really could not be done without you. Thanks to you, we are really building the largest database of genetic and phenotypic information, and it is going to make a drastic impact on our understanding of cats. So thank you all, truly.
All
And then
right,
Jill
we're going to
comes
get started with our Q&A. I need the host to enable my video because I think it was turned off by you all. But we're going to join now and have a conversation with everyone in a Q&A. We've gotten a number of questions in. I wanted to note that some of you that we are answering some of them in written form. So if you can also look at the in the question place. There's answers and what questions were answered, and we'll get going. Hopefully, for some reason, I'm back among you and excited, so you don't have to hear a floating voice. All right, we have about 20 minutes before the end of this conversation, and I'm excited to get going on your questions and share a little bit more. We've gotten a lot of questions from you live. As I mentioned before, we had close to 200 in the registration process on everything from, a lot of specifics about the struggles of your own cats to how are we doing this research to what were the fun things you discovered about fur. So we're going to get to as much as we can. But I think I'll start at first because I'm seeing here a lot of questions about specific things that our cats are struggling from that we really want the answers to. And I know, Elinor, you and I have cats with allergies and have had a fair amount of frustration with the fact that, we go to the vet and there's a lot of guesswork involved. So I guess the question I'd pose is, a lot of what we're doing is foundational research, and I'd love to hear, a little bit about why that's needed and sort of what we're looking at and sort of what it means truthfully about how soon we'll have an answer to why Gus and your cat have chronic allergies.
Thank you very much, Jill. That's a great question. I'm wondering whether we should stop the screen sharing so that all of our heads are bigger for everyone. Because who doesn't want more of our heads, right?
Apologies. I'm still, this is it, as we're going to say 100 times, our first ever webinar.
Yeah. No, I think it's a great question. And I think some of the other questions we've gotten get at why are we asking so many questions about things like coat color and not necessarily about a lot of diseases at this point in time. And one of them is just that things like coat color and striping and things like that are genetically a little bit simpler. And so when you do something brand new, you want to have some things in there that you're pretty sure are going to work. And so we think we'll be able to do some really nice genetics with the kind of physical traits, as we call them. And so that's kind of where we're going to start. I think the other part to it was just that we don't want to ask everybody 1,000 questions about all sorts of really details about diseases if they're not going to be necessarily the best questions to answer. So we're working a lot with veterinarians that specialize in cats. And if any of them are on this webinar right now, I do owe you an email, and I totally know that, to try and actually make sure that when we start asking more questions about cat health, that we're asking the right questions, the ones that are going to get the information we need to push the field forward the most. As Jill said, my cat Lacey is really prone to allergy. She gets skin issues. I have to give her steroids and allergy shots and everything. And she actually had a sibling who died quite young from an autoimmune disease. And I've asked the vet so many times, is there a genetic connection there? Is her allergies related to what was going on with her brother? And they don't know. They can't give me an answer. And so it seems like right now we're at a point where people have many, many questions and there's not a lot of answers out there.
Great. Well, maybe I'll piggyback on that and turn it to you, Kasia. One of the other questions I see a lot we're getting is, when are we going to get results about our cats, and what are those results likely to be? So, do you want to talk a little bit about what we're seeing for like the next, the first round of results, and where we might be going after that?
Yeah, I mean, so first of all, thank you for, contributing without, getting anything back right now. And we are working on it. And as you saw, it's active research. And so I think our first actual return of results we're hoping to provide is going to be around those survey-based results, right? So we talked about the behavior factors or those personalities. And we're hoping to do that later this year. And the genetic research is also, forthcoming. And so, we're aiming to do something around ancestry, shortly after that. Obviously, it's research. It takes time. And so I definitely appreciate everyone's understanding and patience, and contributions to making that happen.
I'm laughing every time Kasia comes up with a timeline in her answer because you can tell how nervous she is. It is a fundamental difficulty of what we're doing, is that the interesting thing about discovery-based science is that we don't know what the answer is, and we're not totally sure how hard it is going to be to get there, which is why it can be very difficult to put hard timelines on things, because we're trying to discover an unknown. But based on things being the process more known versus unknown, we can be a little bit more confident. And so I think that, as you said, Kasia, we should be able to get there.
Great. Well, and one question, we're getting a lot of questions about the survey responses and surveys in general. And one question, well, a couple of different questions I've seen. One is, my answers would have been a lot different if I had answered this when my cat was 5 versus now they're 15. What are you trying to capture? And then also, can we go back and change survey questions or take it over time? What's your approach to that? Elinor, maybe you can start. You've done this a lot.
Yes, this is something we've thought about for years. So just briefly, we never throw out people's answers. We want to keep as much information as possible, and every survey response gets time-stamped. So if you answered the same question twice, we will keep both of those answers, and we could go back through our databases and actually see the change with time, which is actually really interesting scientifically, how traits change as animals age, and also whether some animals that, do the animals that end up living longer actually have a slower trajectory in terms of how they change as they get older, things like that we're really interested in. One of the things we don't do very well yet is what we call longitudinal data collection. We would like to be able to follow animals for their whole lives and learn about them, say every year, actually go back and ask you about them, so we can actually really capture really good information on how they change as they get older. And so one of the things that we're working on developing on the website is better ways to actually do that. And a component to that is the kind of thing we're doing now. If we want you guys to stay engaged in our research and keep on giving us information, we feel like we need to be giving information back to you and actually have it be a conversation over time.
That's great. I know I had fun taking all the surveys, and I'll put in a little plug here for folks to take the surveys. And the more data we have on your cat, the better. One kind of question around sequencing that I see we're getting a lot, and I may have been answered, but I can't see those live, is we do request a charitable donation to participate in sequencing, and that's because we are a nonprofit supported entirely by charitable contributions from foundations and from our community scientists. It is 100% tax deductible because it is a contribution to scientific research. So I wanted to just make sure that is clear. And as Elinor mentioned, it is different from the way we do it is more exploratory and discovery research rather than kind of looking for your cat breed or what have you. Which brings me to, Elinor, a question we're getting a lot is about cat breeds and the difference with dog breeds. And so I'd be curious to maybe you could touch on briefly, what do we know about cat breeds? What do we think we're going to find in the genetics? Sort of where is the state of the science there? And what are we going to be looking at here as we're going forward?
This is a complicated question. With dogs, we dog breeds are very well defined. So about 150 years ago in Victorian England, everybody got very excited about genetic purity and started creating dog breeds. And what they tended to do is they took a small number of animals and they said, these are the golden retrievers. Every animal that has golden retrievers, one of these dogs as their parents is a golden retriever, and no other dogs get to be golden retrievers. It's a very kind of parentage-based definition. And so within a dog breed, all the animals are very related to one another. Cats have breeds. Some of them might be like dog breeds. We suspect some of them might be more like what we in dogs tended to call things like landrace breeds, which are sort of like populations where you define them as being that breed based on what they look like more than who their parents were, especially often with cats, you don't even know who their parents were. And one of the things that's going to come out of the work that Kasia's doing is actually to try and answer that question. When somebody says that their cat is in a breed, what is that actually telling us about their genetics? Because right now we don't know.
Well, that's a great transition. And actually, Kasia, I'll call on you real quick. And, because I know you by training are a population geneticist and did most of your work. Elinor was in dogs, you were in humans. And this is your first time shifting to cats. And I'd love to hear you talk, your specialty really is ancestry, and talk a little bit about what excites you about moving the transition from human population genetics to cats. And what you want to discover.
I'm super excited to be working on cats, and I think it's part of it is because the double-edged sword is that there's so much less work in research in cats than there is in humans. So coming from humans, it's both wonderful because it means we have so much discovery that we're going to be doing. Like, it's really exciting because there's, so little understanding of the sort of domestic cats in the US and around the world, and what they're like. And then on the flip side, of course, it is more challenging because we're really starting from the ground up. And so in humans, there's a lot more infrastructure and resources to really drive your research forward. And we're sort of building that for cats right now as we're going. So it's really exciting because I do think we're going to be really telling the story. And I'm excited to have this at this level of data and resolution that has just not been done before. And telling the story of cats. And who doesn't want to know about the story of cats and how they came to live in our homes with us?
Well, I'm certainly interested in that. And maybe we can talk about the data that's going to help us tell that story. We've gotten some questions on the first sequencing. And so, Chad, I'd be curious, you've done a lot of sequencing process development over your career. What surprised you most about kind of the results of the first sequencing R&D process?
This might sound smug, but the thing that surprised me the most was how well it worked. I've tackled a lot of things in my career that people said, "Oh, that's going to be really hard," or "You're not going to be able to do that," and this was one of them. And it really worked exactly how we drew it up on the whiteboard on day one. So I think the real surprise was for everybody else who didn't think it was going to be doable. For some reason in the field, people were skeptical.
I'm not going to lie. When Chad first told me, he's like, that doesn't seem very hard. I can totally do that. That's not going to be hard at all. And I was like, I feel like Chad maybe doesn't know what he's talking about because everybody else keeps telling me that this is really, really hard. But I have to give it to you, Chad. You totally managed to, you proved me wrong.
So the first conversation I had was to go to a CRO that will be unnamed. And I said, okay, this is what we're going to do. And they said, no, you have to do this from saliva. And I said, no, we're going to do it from fur. And they said, everyone knows that you can't do that. Everyone knows that can't be done. And I'm thinking, yeah, you don't know who you're talking to.
Well, that's great. And I think, we proved it could be done, but I've really enjoyed having a front row seat on the trial and error. And so I'd love, there are some errors. You learn some things. So what was maybe one of those?
Yeah, every process that you develop has hitches. Nothing ever goes as you absolutely expect it. So the first thing that we did was collect the samples in little plastic bags, little Ziploc bags. And that was a disaster because the static electricity made everything stick to the bags. They're really hard to open and close. We had to pick them out with the tweezers. the lesson number one was don't do that. And we had to come up with another way of doing it. The combs were already thought of, but we had to put them in something that we could ship and something that would work in the lab. And I think it was Benoit's idea to use the standard 15 mil laboratory tube. So that worked very well. The other problem that we had was, I think I mentioned this when I was talking, was that we got so much DNA the first time we did it that it actually was impossible to QC, and we had to dilute it and dilute it and dilute it until we figured out what was wrong. Oh, the problem is we have too much DNA. And so we were so concerned that we weren't going to get good yields that we overdid it initially.
That's great. Well, and
That was a fun problem to have.
it is a fun problem to have. And I guess we're getting close to time, so maybe I'll turn it back to Elinor for a couple of last questions. One thing we've gotten a lot is sort of what are the goals of this project, in 2 years, and sort of what we hope this will bring about in cat science over the next 10, 20 years. I've heard a lot of reference to the Human Genome Project. So I'd love for you to touch and kind of end with, where are we hoping to go in the short term and then in the long term?
No, thank you, Jill. It's a challenging question because, as I mentioned earlier, as a scientist, you try not to put too many predictions on things because sometimes things don't work. But what we've designed the project in a way to try and make sure that we're going to be discovering things both in the short term and in the long term. So I'm hoping that within a few years, we're going to have figured out some of the basic coat striping genetics in cats. Cats have an absolutely phenomenal range of coat patterns, as Kasia and I both discovered when we thought we should ask some questions about this, and went and looked into it and discovered that there's like 4 different kinds of tabby striping or something like that. I had no idea how complicated it was. And all of that gets set up very early in embryo development, and nobody really understands all the intricacies. There's some very nice work showing that it has to do with when genes get turned on and turned off. And so hopefully within a few years, not only on a scientific level are we going to understand more about the origins of that, but we will actually be able to do some fun kind of genetic things, like sort of make predictions about what a cat's going to look like based on its genetics. You already know what your cat looks like, so maybe that'll be less impressive, but I do think it's cool when you can do it from genetics. And hopefully from all of our survey data, we might actually be able to start having more of a sense of which traits are actually being controlled more by genetics in cats and which diseases are really the ones that we want to be looking at. Things like being able to say something about disease are a much longer term proposition, which is why we kind of always keep talking about this being a longer term project. We need to find things that are associated with the disease in cats. That's the sort of piece that I talked about. But in order to be able to make a prediction about whether a cat is at risk of a disease from their genetics, we need to do a whole other study after that, where we get another set of cats and ask whether doing the genetic test actually does predict whether they're going to get the disease or not. We want to make sure that the information that we're giving back will actually tell you something about that cat's disease risk. So you have to kind of do both parts of that project before you can actually actually use it in sort of a clinical sense. So that's the 2 pieces. We're kind of thinking about both of those kinds of things in parallel, the scientific discovery, but then also how do we actually take the next step after that and develop it into something that will actually improve cat health. And that's a longer term goal. But as you know, I've talked to you a lot about this, Jill, but it's the only way we can get there, right? Like right now, before Darwin's Ark, we didn't have a path from where we are with what we know about cats and cat genetics now to actually being able to do anything useful clinically. And this is starting us on that path. And for me, that's really exciting. Like I can see how we can get there now, which is fantastic.
Well, great. Elinor, I'm going to ask you one last extremely hard-hitting question that we got from a participant, which is where you got your cat wheel, or what brand that is. So I take it there's some people who'd like one as well. If you happen to know it.
Yeah, it's a wonderful cat wheel. Unfortunately, the company went out of business, so you can't actually get it anymore. So now I'm like paranoid that it's going to like break down, and poor Lacey is going to be bereft because she uses that like all the time. She likes to show off. If she gets on her cat wheel, she likes it if you look at her and admire her and tell her how amazingly wonderful she is, then she runs even faster on the cat wheel. It's hilarious. But I highly recommend cat wheels. If you have a very active cat, they're like a game changer. They really love using them.
Well, great. Well, thank you. And thanks to everyone for all of the questions. We've got a lot. And as I mentioned, we didn't get to them all or nearly close to them all. We are going to be finding other ways, newsletter, blog, future science talks. We got a lot of questions on behavior. We got a lot of questions on health. We'll be talking to the scientists on our team that are behavioral specialists and veterinary specialists and figuring out ways we can have future science talks that are really leaning into some of the questions that you've asked today. And then also continuing to update you on our research. I did get one question that I'll just answer briefly. We've got 19 surveys now, but the whole plan is to continue to add more surveys. As Elinor mentioned, we started a lot with traits, physical traits and behavior, and we are going to be adding health surveys and other surveys as they go. So please keep your eye out for emails and continue your participation. It really means a lot. After this, we're going to send the recording of this if you'd like to view it or share it, and also a brief survey about this webinar. So we'd love your feedback. Please, this is our first webinar, as we've made clear a few times, and so we don't want it to be our last. And we'd really enjoy knowing what you liked and what you'd like to see more of or less of in future events. Thank you so much for your amazing role in Darwin's Cats. And I look forward to talking with you again soon. Thank you all and have a really great day.